Good afternoon, everyone. So, today we'll talk about gene therapy. But before that, let us define first what is gene therapy. So, gene therapy is a method to cure or treat genetic related illnesses. And these illnesses are related to defective genes. Meaning to say, defective genes, mao siya ang mga genes nga imperfect or wala na siya nag-function fully. And these type of diseases are not curable by traditional methods. When we say traditional methods, kay mao ni siyang mag-take og medicines nga mapalit nato sa pharmacy. So kani siya nga mga sakit is dili maulian by taking medicine only. And there are several examples of genetic related illnesses. So naa ang eye disorder, naa ang cancer o ang Down syndrome. Daghan pa kaayo nga mga examples, but the first two are not purely genetic, but naa lang sila'y genetic component. And atong 2015, ang researchers sa Medical Harvard School and Children's Boston's Hospital kay stated that they were able to restore the basic hearing of a deaf mice by using gene therapy. So obviously, effective jud ay ang gene therapy. Kay imagine, ang deaf mice na restore iyang sense of hearing, kay gigamitan man siya inani nga therapy. But, ang atong 1971 na ana di ay human gene therapy. Naana sila ay patient nga naay genetic illness nga ganahan siya ma-remove ang katunga disease. So, nangita sila foreign DNA. When we say foreign DNA, is dili DNA sa patient, but DNA sa other organism. And then, katunga DNA is gi-insert sa patient, hoping nga ma-eradicate or ma-remove tong diseases. And without a doubt, gene therapy is the most promising. Why? It is because na possibility nga ang mga people nga naay genetic illnesses kay naapay chance nga maulian by using this therapy. However, it is still complicated and challenging regarding the safety and the side effects nga ma-acquire sa patient.
Replacement. So, replacing a mutant gene with a healthy one. So, replacements explains that, or it means that this approach involves removing or bypassing a defective one or a muted one. So, muted genes and inserting normal healthy versions of this so that the body can function properly against. So, for example of this, so in service combined with immunodeficiency or SCID, so the doctor replaced the muted genes or responsible for immune systems failure with a healthy one, so that allowing the patient's immune systems to work normally. Inactivations. So, inactivating a genes that is working improperly and causing harm. So, inactivations. So, this approach turns off or disables a harmful genes that is causing disease. Can prevent it from producing damaging effects in our body. So, for example of this. So, in some cancers, a genes that cause uncontrolled cells can be inactivated to stop tumor cells from multiplying.
One of the basic approaches in gene therapy is adding or introducing a new gene into the body. This new gene is designed to help the body fight a disease more effectively. So what does it mean? Some diseases happen because the body does not have a certain gene needed to function normally. To solve this, scientists insert a new working gene into the patient's cells. This new gene helps the body perform a task it couldn't do before, such as fighting infections or producing a missing protein. How is the new gene delivered? Because if a gene cannot just enter the body on its own, scientists use carriers, also called vectors. Most common vectors is viruses. The harmful parts of the viruses are removed. The virus is genetically modified to carry the healthy gene. The virus then delivers this gene into the patient's cells, not to cause disease but to help fix it. Think of it like the virus becomes a delivery truck bringing a healthy gene package into the cell. Why is it helpful? introducing a new gene can help the body fight a disease. Also cells to produce protein the body was missing, strengthen the immune system, reduce symptoms or stop diseases at its root. For example, this method can be used for immune system disorders, help the body produce missing immune cells. Certain cancers gene produced to attack cancer cells. Inherited diseases caused by missing or defective genes. Into the body, the gene cannot travel or enter the cells by itself. It needs help getting inside the right cells just like a package needs a delivery service. This is where carriers, also called vectors, come in. What is a vector? A vector is a tool or vehicle used to transport the new, healthy gene into the patient's cells. Think of it like the vector is a delivery truck and the new gene is the package it carries. Without a vector, the gene would not reach the target cells and would not work effectively. Why are viruses used as vectors? Viruses are the most common vectors used today because viruses are naturally good at entering human cells. They can deliver genetic materials inside the cell. Scientists can modify them so they no longer cause disease. In gene therapy, the harmful parts of the virus are removed. The virus is genetically changed, it becomes safe, and it is loaded with normal human DNA. So instead of causing illness, the modified viruses delivers the healthy gene into the cell. How does it work? Scientists take a virus and disable its ability to harm the body. They insert the new, healthy gene in the virus. The virus enters the patient's cells. Then, the new gene is released and starts working to help the body fight the disease.
I'm Priya and I will be discussing and explaining the two types of gene therapy. So, gene therapy is a medical technique where scientists change or fix genes inside our bodies to treat diseases. So, there are two main types, which is the somatic gene therapy and the germline gene therapy. So, let's start with somatic gene therapy. So, this type of gene therapy focuses on body cells. These are cells that do not make babies, like muscle cells, skin cells, or bone marrow cells. In somatic gene therapy, doctors change or repair the genes inside this body cells to help a patient recover from the illness or an illness. For example, if a person has a genetic disease, doctors can fix the gene in their body cells so that the patient become healthier. But here's the important part: the changes only help the patients themselves. These changes are not passed down to their children. So, somatic gene therapy helps a person get better, but it does not affect the next generation. So, second, we have germline gene therapy. So, this type focuses on germ cells. These are the cells that produce babies, such as sperm cells, egg cells, or the original cells that form in embryo. So here the genes inside these germ cells are modified. What makes germline therapy different is, so any genetic change moved to these cells will be passed on the person's future children. So that means the next generation will also carry the corrected or modified genes. So, germline gene therapy doesn't just help one person, but it also affects future generations as well. So, overall, somatic gene therapy changes genes in regular body cells and helps the patients only. But changes are not passed to children. So the germline gene therapy changes genes in sperm, egg, or early embryo cells. So the changes are passed to future generations. So both types aim to prevent or cure genetic diseases, but they differ in how far the affects each other. That will be all.
Now we tackle about stem cell gene therapy. Stem cell gene therapy is a medical approach that combines two powerful fields, stem cell therapy and gene therapy. It uses the unique abilities of stem cells to repair or replace damaged tissues, while gene therapy corrects or modifies faulty genes inside the cells. So together, they aim to treat diseases that were previously incurable. So what are stem cells? Stem cells are known as mother cells because they can become almost any types of cells in our body, like blood cells, bone cells, muscle cells, or even brain cells. What makes stem cell very special is that they can renew themselves, meaning they can make more copies of themselves. They can also change into other cell types depending on what the body needs. Because of this ability, stem cells are used in gene therapy, where doctors fix or replace faulty genes inside the cells. When the corrected stem cells are placed back into the body, they help create healthy and normal cells which can treat certain diseases. Now, for example, imagine a child who easily gets tired and cannot play normally because they have a blood disorder. So the doctors takes the child's stem cells, correct the damaged gene, and return the fixed cells to the child's body. So eventually, the child begins to produce healthy blood cells and can live a normal and active life. This shows how stem cells gene therapy can help real people. This is why stem cells are very useful in treating diseases where tissues are damaged or cells are not functioning properly. Next, let's talk about where stem cells come from. One important source of stem cells is called embryonic stem cells. These come from a 4 to 5 day old embryo called a blastocyst. Most embryonic stem cells come from IVF clinics or in vitro fertilization. During IVF, many eggs are fertilized outside the body to help a couple have a baby, but usually only one embryo is used for pregnancy. The remaining embryos are often donated for research with the parents' permission. Scientists use these donated embryos because embryonic stem cells can turn into any type of cells in the human body. This makes them very useful for studying diseases and developing new medical treatments. For example, imagine a couple trying to have a baby through IVF. Several embryos are created, but only one is implanted into the mother. Instead of throwing away the extra embryos, the couple chooses to donate them. So the scientists then use the stem cells from these donated embryos to study how to repair damaged organs or treat diseases, which may help many patients in the future.
Somatic adult stem cell ay mga stem cell na makikita natin sa iba't ibang tissues ng katawan tulad ng brain, bone marrow at liver. Pagkatapos ng embryonic development, ang kaya nilang maging limitado depende sa tissues kung saan sila organically galing. Halimbawa, kung galing sila sa bone marrow, hindi sila pwedeng maging brain cells. Pero kahit limitado sila, may special ability pa rin sila na kaya nilang mag-divide o dumami at mag-self-renew, mag-repair ng sarili. So dahil dito, nakakatulong sila sa pag-regenerate ng tissues o minsan pati buong organ kung kailangan ng mag-repair. Where do they come from? So galing sila sa embryos o sa adult tissues tulad ng bone marrow, liver, brain at skin. Ang embryonic stem cell ay pwedeng maging almost any cell type. So habang ang adult stem cell ay limited lang sa tissues kung saan sila galing. So ang good versus bad. So kapag ginamit ang stem cell para ayusin ang sakit o damaged tissues. Example sa bone marrow o transplant para sa leukemia. So sa bad naman is kapag ginamit sila para gumawa ng genetic changes na hindi para sa tulad ng pag-modify ng traits sa halimbawa sa eye color, sa height, sa intelligence, ayun, Ito ay risky kasi pwedeng magdulot ng side effect at may ethical issues.
Normal versus disability. Who decides which traits are normal and which are a disorder that needs to be fixed? First, let's talk about the question, who decides which traits are normal and which one are considered a disorder? When we think about genetic condition, we often assume that everything different must be fixed. However, not all traits that seem like disorders are actually harmful. Some traits are simple part of human society. For example, communities such as people who are deaf, autistic, or dwarf don't always see their condition as something that needs a cure. For them, it is part of their identity and the culture. Let me give you an example. Imagine a couple who are both deaf. With gene therapy, doctors now have the technology to make sure their baby can hear. But what if the parents don't want that? What if they want their child to be part of their deaf community? This creates an ethical issue. Should society force them to choose the normal trait just because most people think it's better? Or should parents be free to decide? So, the main point here is that defining what is normal is not always a scientific decision. It is also a social and cultural decision. Next, let's move to the second issue, cost and availability. Gene therapy is one of the most advanced and expensive medical technology today. It requires special labs, high-level experts, and very complex procedures. Because of this, treatment can cost millions of pesos. The problem is, if only the rich can afford these genetic treatments, then healthcare becomes unfair. For example, a child with rare genetic disease like spinal muscular atrophy may need gene therapy to survive. A rich family can pay for it, so their child lives and becomes healthy. But a poor family might not be able to afford it, so their child does not receive the same choice. Over time, this can create a society where wealthy families are not only richer, but also healthier and genetically advantaged. Meanwhile, poorer families fail behind. This raises an important question for our future. Should life-saving technology only for those who can afford it? Or should everyone have equal access?
Gene therapy has the potential to cure diseases and improve health. But if it becomes widely used, it could also affect how society views people who are different. So first, gene therapy might create pressure to be perfect. If most people use it to remove certain traits, those traits might be seen as unwanted. So second, differences such as disabilities or learning challenges might become less accepted simply because they become less common. So the third is those who cannot afford gene therapy or choose not to use it might be judged or discriminated against. People could wrongly think they should have fixed their genes. In short class, widespread gene therapy could accidentally make society less inclusive. Instead of celebrating diversity, we might start treating differences as problems. This is why ethics and equal access are important when using gene therapy. Enhancement in gene therapy means using it not just to treat diseases, but to improve normal traits in people. For example, someone might want to use gene therapy to become taller, smarter, or more athletic. So this raises big questions. First is fairness. If only some people can afford enhancement, it could make society even more unequal. The second is pressure. People might feel they have to enhance themselves or their children to keep up. The third is identity. So, changing basic traits could make us question what it means to be human and unique. So, while gene therapy class can help treat serious and illnesses, using it to enhance normal traits is much more controversial. Society has to decide where to draw the line between helping people and changing what it means to be human.